During prophase 1, the complex of DNA and protein known as chromatin condenses to form chromosomes. A large structure called the meiotic spindle also forms from long proteins called microtubules on each side or pole of the cell. Between prophase 1 and metaphase 1, the pairs of homologous chromosomes form tetrads. In metaphase 1, the homologous pairs of chromosomes align on either side of the equatorial plate. Each daughter cell is haploid and has only one set of chromosomes, or half the total number of chromosomes of the original cell. Anaphase 1. This separation means that each of the daughter cells that results from meiosis 1 will have half the number of chromosomes of the original parent cell after interphase. Also, the sister chromatids in each chromosome still remain connected. As a result, each chromosome maintains its X-shaped structure. Telophase 1 is that phase when the chromosomes have finished moving to opposite ends of the cell. This will then be followed by cytokinesis, producing two daughter cells. Thus, after meiosis 1, each cell would have a haploid set of chromosomes, but each chromosome would still be comprised of two sister chromatids. During prophase 2, the chromosomes condense and a new set of spindle fibers forms. The chromosomes begin moving forward the equator of the cell. The spindle fibers pull the separated chromosomes toward each pole of the cell. Finally, during telophase 2, the chromosomes are enclosed in nuclear membranes.
These gametes divide and get half the number of chromosomes. This is important because it allows both parents to pass on half the genes when reproducing. When prophase 1 occurs, the chromatids line up next to their homologue. This forms tetrads. When tetrads form in homologous pairs, parts of the chromatids may break off and attach to chromatids beside them, crossing over. This results in variation of the offspring because new gene combinations are made. In a single trait Punnett square, if there were to be a homozygous parent with blue hair and a homozygous parent with red hair, and blue was the dominant allele, in this case, all the blue alleles would be dominant over the red alleles, and all the offspring will have blue hair as phenotype. However, in the genotype, there will still be a hidden allele for red hair. If the parents were heterozygous, only 75% would have blue hair in the phenotype, and 25% would have red hair. In the genotype, 50% would be heterozygous, 25% would be homozygous for blue hair, and 25% would be homozygous for red hair. In codominance, both alleles are expressed. They are neither dominant nor recessive. The alleles work together rather than one trait dominating the other. If we have a homozygous white rabbit and a homozygous gray rabbit, in codominance, the offspring will be heterozygous, but since these are codominant alleles, the offspring will have white fur with gray spots, expressing both alleles. In incomplete dominance, neither allele is completely dominant nor recessive. For example, if there were a homozygous white flower and a homozygous black flower, their offspring would all be heterozygous and would result in both traits expressing incompletely, resulting in a gray flower. There are 4 blood types, types A, B, AB, and O. This is because there are 3 different alleles, IA, which is dominant, IB, which is also dominant, and I, which is recessive. IA and IB are codominant, making AB blood. A blood carries an A antigen and has a genotype of either IAIA or IAI. B blood carries a B antigen and has a genotype of either IB/IB or IB/I. AB blood carries both A and B antigens with a genotype of IA/IB. O blood carries no antigens and has a genotype of II. Blood can also be positive or negative, positive being dominant over negative. In a heterozygous mix, the phenotype of the offspring would be 75% positive and 25% negative. In the genotype, 25% would be homozygous positive, 25% would be homozygous negative, and 50% would be heterozygous. So there are actually 8 blood types. Let's say that there are 2 parents, one with a genotype of IAI+, and the other with a genotype of IBI negative. If we were to make a Punnett square for them, 25% would be AB positive, 25% would be B positive, 25% would be A positive, and 25% would be O positive. In their genotypes, 75% would carry an allele for O blood, and they would all be heterozygous positive, carrying a negative allele but not showing it in their phenotype.